just when you thought it was safe, here I am back again. This is the second part of that screencast I gave just yesterday that sort of summarized everything and included some Gizmo information, etc. So I'm going to start off here. You saw this already, so it's looking at the structure of the mitochondria. This is a mitochondrion, and here we see an electron micrograph of that. We mentioned already this, so we said that it's in the mitochondria that both the link reaction and the Krebs cycle, or the citric acid cycle, take place. And we went through this idea of here, this is the product of the link reaction, how acetyl-CoA with its 2 carbons joins with oxaloacetate with its 4 carbons. We have a little thing going on right here. We form citrate, and then citrate has 6 carbons, and then we sort of follow the cycle around until eventually what we've done is take away electrons via these electron carriers. So we have NAD getting reduced into NADH, and then we have FAD, which is getting reduced into FADH2. We make a little bit of ATP here. We see GTP. This gets converted to ATP, and then we go from there. And from there is where we're at. So from there, what we get are what's happening with the NADH and the FADH2. So this is just showing one piece of it. But what happens with this is NADH will come to the inner, so here we have the intermembrane space. So this would be the outside membrane of the mitochondria. And then embedded in this inner membrane, remember those like weavy parts of the membrane we called cristae. So this is the interior membrane. We have these carriers. These are part of an electron transport chain. So these are embedded in the membrane. And when the NADH is in proximity, it's this carrier molecule in this electron transport chain will take the electron from NADH, electrons. And because this thing here has a higher affinity for electrons, so it takes them off of NADH, and it also takes the hydrogen proton, and the proton gets pumped out into this space. So here you can see the protons being pumped out into this space. Now, FADH2 also does that. It comes in a little bit later in this game. But at the end, the electrons just get bounced from one thing to another, these proteins embedded in this membrane that are taking the electrons, passing it from one to the next, meaning that each one that's next door that's taking it has a higher affinity for those electrons. And then at the end, the ultimate electron acceptor is oxygen. So here finally we get to oxygen. And oxygen is going to pick up those electrons. And what we don't see here is that we're really not looking at O2 now. We're going to be looking at half of an O2, right? So not the molecular form of oxygen, but the atomic form. And it's going to join with some hydrogen that are free in this matrix, in this inside space here, to form water. So oxygen is important because oxygen is the most important receiver of all of these electrons, and when it does, it forms water. But what's also important is that the electrons are kind of just giving the energy for this whole thing, but really what they're giving the energy for is pumping these protons out here into the space. So now we're going to get this buildup of proteins. We're going to get a higher gradient, remember that, of protons in this upper area in this space. And what's going to happen when these protons start accumulating out here is the protons are going to move then through a new molecule embedded in the membrane, has an ASE ending on it. You guessed it, it's an enzyme. So this is called ATP synthase. So I am going to go here, so because that's just, this is really from OpenStack. So here you can see NADH electrons are going to get passed along this complex. Ions are going to get pumped out into this intermembrane space. FAD, here it is again, but it's starting a little later in this process, doing the same thing though, passing its hydrogen out here, the protons rather, into that intermembrane space. They start to build up. At the end, we have this half of an oxygen that joins with two hydrogens and forms water. Okay, so the electrons are important in this process. They get pulled in this direction. And then, this kind of doesn't really show it, I think, well enough to say that up here we're going to have lots of hydrogen protons that have gotten pumped into this little space above, and they're going to start moving down their concentration gradient. And as they do, this thing spins, and the energy from that flow of electrons or protons down this ATP synthase, it's going to provide the free energy necessary to put that free phosphate onto ADP and now form ATP. So this is a very important process. We are phosphorylating ADP via this oxidative way. So this is oxidative phosphorylation. Here you can just see it again. It's a little bit more expanded view. I think this is kind of helpful to kind of study this diagram. And then finally, what happens, if you remember in the POGL too, what happens if you don't have enough oxygen? Well, fortunately, if you are exercising a lot and you don't have enough oxygen, we have kind of a bypass that we can do. So here, remember, is glycolysis. Glycolysis is taking that glucose and changing it into these two pyruvates. And while it does that, it's creating these NADHs, right? So this is the normal process. So now we have pyruvate. But if there isn't any oxygen, pyruvate won't go into the link reaction, into the Krebs cycle. And so you just get this accumulation of pyruvate, and you get an accumulation of NADH that then can't make any more ATP. So we got to free up NADH. Free NADH. And the way we do that is take a little detour and take pyruvate now and form something called lactate. And in doing that, notice what happens here. That NADH that was made now is oxidized. So we can take the electrons and the hydrogen off of it. So now it's available to go back up and do this again. So we can make a couple of ATP. We can continue to make a little bit of ATP in the cytoplasm, even when we don't have oxygen present. So this is the lactic acid form of fermentation. There's another one that your POGL shows you, which is similar, that bacteria use, and that is the alcoholic fermentation. So here we go. This gets into some of the Gizmo stuff, which is fascinating stuff. And what it does is it's giving you that scenario about the poisoning of the CIA agent, and it says we got to figure out what poison was used. And we can figure out what poison was used based on what part of this whole cellular respiration series of things, and what's happening at what step of that. Because each of these poisons act at a different location. So here's the first one. They give you this deoxyglucose, what you see here, this 2-DG, as a potential poison. It looks like this. Notice it looks so much like glucose. It just has something substituted here. Remember, it's deoxy. So look at, this isn't a hydroxyl group. It's just a hydrogen. But if this is the poison and it enters the cell through this GLUT1 pathway, so it comes through here. Normally, if this was glucose, then we would have hexokinase that would convert it into, if you remember, glucose 6-phosphate. But this gets inhibited right here. And that ultimately stops glycolysis from being completed and would result in cell death. Okay, this also, I'm going to get back to talking about cancer cells too. So this says cancer cell here. That's not a part of the Gizmo thing. I'll talk about that again. As a matter of fact, I might even talk about it now. So interestingly, cancer cells operate differently than normal cells. And one of the things that they do is when they do cellular respiration, they really primarily just do the glycolysis part of it. And so what happens then is the cell is seemingly starved for energy because it isn't doing the rest of it, where usually more ATP comes down the line from the parts of cellular respiration that take place in the mitochondria. So if it's not doing the rest of it because it's getting messed up early in the game, we get a buildup of lactic acid for one thing. And what happens is the cell is like the cancer cell is trying to get as much energy as it can. So it consumes far more energy than the normal cell. It takes more glucose. It's kind of stealing glucose from your normal cells. So that first step of a cancer cell, if we could inhibit it using a toxin or poison to make just the cancer cell get messed up when it was, so that it was sort of crippled, then that would be a good potential treatment. Another one that Gizmo's talked about is arsenic. Now, you may have heard of arsenic, or maybe not. Arsenic, I think, used to be put into like rat poisoning and things like that, or maybe I think I'm sure on that one. But anyway, there's also a great book called Arsenic and Old Lace, which is a murder mystery. I think might be an Alfred Hitchcock if you're curious. But anyway, arsenic is another one. Arsenic doesn't interfere with the glycolysis at all. Arsenic interferes with the link reaction because it's going to bind to the enzymes early on, and just when they go into the mitochondria, into the one that converts pyruvate into acetyl-CoA. And then what happens, if it binds to that enzyme, then pyruvate doesn't get converted, and that just gums up the works. You start getting a lot of pyruvate building up. Okay, so that's kind of, and then the Krebs cycle effectively gets stopped. Cyanide, another lovely material, poison, its main way that it affects things is in the electron transport chain. So it's going to attach to one of the last enzymes in that chain and basically stop that electron from going any further. And if you can't move the electrons along that chain, then you don't pump those hydrogen ions out into the intermembrane space, and that's a bad thing because then you can't generate ATP. So just to kind of show you, this is kind of where cyanide could interfere with this right here, and then that's bad news. And so these are, I just pulled this from this website or from the internet just to show you. So there are lots of poisons and toxins out there, and a lot of them act directly on some of the enzymes of these different stages of any metabolic process. But here we're looking at cellular respiration. So these things are going to interfere. Here's carbon monoxide, also a bad thing to have high exposure to because of what it messes up here on the electron transport chain. So lovely topic, important though, and kind of gives you some understanding then of what poisons and toxins actually do when we say they're toxic or they're poisonous. And that's it.